Everyone, thank you for coming to this session. My name is Bernard Siegel. I'm the Executive Director of the Regenerative Medicine Foundation and the founder of the World Stem Cell Summit. And in this session, we're going to have award-winning research presentations. This was supported by AlphaMed Press. And as you know, AlphaMed Press is our partner, and the official journal of the Regenerative Medicine Foundation is the high-impact journal, Stem Cells Translational Medicine, that is, whose editor is Dr. Tony Atala, Anthony Atala at Wake Forest Institute for Regenerative Medicine, who's also a member of the board of our organization. Our first award-winning presentation that was honored by the journal Stem Cells is Gunas Uzer, Assistant Professor at Boise State University. Gunas, please give your presentation.
Okay. Thank you for the kind introduction. Okay. Is my slides are up? Right. Mm-hmm. Okay. Yep. Okay. Okay. Thank you all for coming. So I'm a kind of an engineer who turned cell biologist. So I'm going to talk about how connectivity of cells and the mechanical signals regulate stem cell biology. So we've been told to exercise when we are young, right? And then we also told to exercise when we are older, and we're supposed to do all these other exercises in between to stay healthy throughout our life. But before we get to the sort of like what the mechanical signals mean for the stem cells, I would like to start introducing or defining what this link complex means. Link complex stands for linker of nuclear skeleton and cytoskeleton. And really, if you have a cell that's attached to a surface, you're going to have three basic structural elements. You're going to have a nucleus that sits in the middle, which is denser and stiffer than the rest of the cell. And we also have the cell membrane that encloses the cellular structures. And we also have the cytoskeleton that connect these two elements together. So if you look at a single cytoskeletal element, what you are going to see is that this is actually a continuous structure. On the left, you have extracellular matrix. You have integrin signaling integrins that binds to the extracellular matrix, forming these mechanosensitive focal adhesions. And then these focal adhesions kind of sequester actin cytoskeleton. The actin cytoskeleton then goes in and then binds itself to the nuclear surface. At the nuclear surface, actin is connected by a protein called nesprin. This is a giant protein that protrudes out of the nuclear surface. And Nesprin is kind of anchored into the nuclear surface through a protein called SUN. And in this way, this protein complex couples the nucleus to the extracellular matrix, hence the name, the link complex. Again, the Nesprin binds to the actin cytoskeleton, and then SUN binds to the intranuclear lamin AC network, basically coupling the nucleus to the outside world. So I'm going to talk about today how this complex plays a role in mechanosensitivity and signaling of mesenchymal stem cells. So we know that the mechanical signals are important for maintaining the musculoskeletal system. And just like our muscles, our bones are also able to respond to these mechanical challenges. This could be either a positive regulation or a negative regulation. In terms of positive regulation, it's known that the playing arm of tennis players are much stronger compared to its non-playing tennis arm. And we also know that astronauts lose about 2% bone mass in space per month. So it becomes a real challenge bringing the astronauts back to Earth without giving them extensive rehabilitation. So this ability of bone cells or bones to kind of adapt to these mechanical challenges come from bone cells themselves. And if you take a cross-section of a bone, you will see there's a cortical bone, trabecular bone, and a bone marrow. And if you look a closer look, you will see a number of populations that are actually responsive to mechanical challenges. One of them is osteoblasts. They are the cells that actually make bone. We have osteoclasts, they resorb bone, and these two cell populations are connected to each other with a process called remodeling. And in 10 years from now, all the bones in your body is going to be remodeled by this process. We also have osteocytes. They are the osteoblasts that get trapped in the bone matrix. And again, we have the mesenchymal stem cells. So these are the cells, the progenitor cells for bone, muscle, cartilage, and fat. And since they can also differentiate into fat, we have plenty of fat cells in the bone marrow as well. So the topic of the talk today is how mesenchymal stem cells are regulated by mechanical challenges. And it's been known that if you exercise and a healthy lifestyle, your mesenchymal stem cells is going to be osteogenic. And in the converse, if you are not exercising well or you're in space, for example, your mesenchymal stem cell is going to be more adipogenic and less osteogenic. So in laboratory or preclinical models, we use exercise or exercise analogs to kind of look at how these mesenchymal stem cell fate is regulated. Here's a recent study that we published that we show that with the mice exercising with a training wheel about 6 weeks, increases the bone mass and also at the same time decreases the fat in the bone marrow. So in cellular level, to study this, we use sort of these analogs of exercise and give cells these mechanical challenges. And one of them is the low intensity vibration, or I will call them LIV for the rest of the talk. And these are the signals that we apply anywhere between 30 hertz to 100 hertz and about 1 G magnitude. And in both preclinical, clinical, and cell level models, we have shown that the application of vibrations can increase the bone mass and decrease fat. And similarly, high magnitude strain is also another method that we use. We use this anywhere between 0.1 to 1 hertz with the 2% stretch of the membrane, and we also, others have been shown that it increases bone modeling. And when we look at fat differentiation of mesenchymal stem cells in a petri dish, we'll see that both LIV and strain can decrease the adipogenic commitment of mesenchymal stem cells, as shown by the well-known fat markers adiponectin, AP2, and PPAR-gamma. So one of the other things that we look at is how these signals are sensed at the cellular level. And one of the things that we have found, as I mentioned earlier, these focal adhesions are these mechanosensitive signaling platforms that exist at the attachment sites of the extracellular matrix. And we have shown that both LIV and strain can activate these focal adhesion kinases at the focal adhesions and also result in cytoskeletal remodeling of the actin cytoskeleton, evidenced by the activation of ROE, which is the chief mediator of cytoskeletal remodeling in these cells. We also look at how these cytoskeletal changes happening using imaging techniques. Here is a control cell. You see green is the actin cytoskeleton, and blue here is the nucleus. And here is the response to strain, response to LIV. I would like to get your attention to this area where after vibration we have this F-actin accumulation at the perinuclear area. And in response to strain, what we see is that these are these stress fibers that are crossing across the cell. So one of the questions that we wanted to ask was how this connectivity within the cells can change or affect these mechanosensitivity. And we wanted to look at the role of link complexes in kind of mediating this. Link complex is a two-part protein complex. The nesprine binds to the actin cytoskeleton, and some proteins bind to the intranuclear lemon network, connecting the nucleus to the outside world. Here's a cell with or without link complexes and how they look. In a control cell, again, you see this green actin cytoskeleton, and the nucleus is in the middle. And you might see the actin cytoskeleton crosses over the nucleus and it kind of pushes it down, making it more flat. In a link deficient cell, and we accomplished by this siRNA against the SUN protein, effectively disanchoring the nesprine from the nuclear surface, and you see that the nucleus is more spherical now, and it has no actin cytoskeleton connections to it. When we look at the structural parameters of the nucleus, we see that the nuclear height is increased, nuclear area is decreased, but the nuclear volume remains the same. So when we look at these in cells without functional link complex, what we have found is that in the absence of the link complex, there was no activation of mechanosensitive pathways. We did this for SI-SUN treated cells and also domain-negative cache treated cells, which is the binding protein or binding complex between the SN and nesprine, so effectively doing the same thing. In both of these cases, we found that the LIV signals require a link nucleus to kind of respond to these mechanical challenges. So I would like to switch gears a little bit now and talk about how these signaling events or mechanical signaling events affect the cellular signaling to kind of change the cell fate in mesenchymal stem cells. In addition to the FACT signaling that we have just shown you, when we exercise, there's other signaling events that happen in the cell, and one of them is the activation of AKT. And AKT goes ahead and inhibits the GSK3 beta, and as you know, GSK3 beta is a part of this degradation complex that actively degrades beta-catenin. And beta-catenin is the chief mediator of Wnt signaling pathway, which is known to regulate the regeneration and development of musculoskeletal tissues. So when mechanical signals increase the beta-catenin in the cytoplasm, beta-catenin goes in and enters through nuclear pore complexes to activate the gene's targets. When we get old, however, not only our MSCs become more adipogenic, but their mechanosensitivity is also decreased. We don't have to wait for years, however, to have insensitive MSCs. In a pathology case of aging called progeria, patients again show reduced mechanosensitivity and increased adipogenesis. And one of the etiologies of progeria is this misshapen nucleus. Here's a control cell, and here's a progeric cell. And we know now that the problems in the nuclear structure come because of the mutations in the lemon network. But now we are also learning that these link complex mutations also play a role in the phenotype of this. So hopefully in this second part of the talk, I will convince you that this connectivity within the cell is kind of important, regulating the beta-catenin signaling, and give you a little bit information about what we're trying to do next. Here is a link deficient cells and link deficient animals, and you see, I showed you before in the presence of SI-SUN, which is a kind of it disrupts the link complex. We see an increased adipogenesis, and at the same time, with the link deficient mice, we see a decreased bone mass, suggesting that this link complex might actually play a role in how beta-catenin signaling is regulated in cells. And we wanted to ask this question. And indeed, what we have found was that in the absence of link complex, we have less beta-catenin in the nucleus, and even mechanical and pharmacological inhibition of beta-catenin is inhibited in the absence of these link complexes. So again, just for the time brevity here, that we started with this link complexes that connect the nucleus to the outside world, and we have shown that the application of mechanical force can increase the FAC activation and can activate ROA. In this way, they actually produce these cytoskeletal structures around the nucleus, perhaps exerting forces to the chromatin directly and change the stem cell fate. And we also shown that these link complexes are intimately connected with the nuclear pore complexes and may play a role in the beta-catenin inward transfer. And one of the things that I didn't show you, we also showed that in the absence of the lamin AC network, link complexes still can exert forces on the chromatin and change the adaptogenic commitment of mesenchymal stem cells. In this way, we are currently studying how these link complexes are regulated mechanically or otherwise, and we think these regulation of link complexes or dysregulation of link complexes play a role in the cases where we have the when we had the reduced mechanosensitivity and beta-catenin signaling in cases such as aging, bed rest, or microgravity, or we can utilize these mechanical challenges or regulation of link complexes in cases like tissue regeneration as well as in bioreactors. With that, I would like to thank all the funding mechanisms that support our work, and if you have any questions, I will be happy to answer. Thank you. Okay. Thank you.
Thank you, and safe travels as well. I'd like to introduce Dr. Thorsten Dopner, University of Göttingen Medical School, who received an honor for his work from the journal, the official journal of the Regenerative Medicine Foundation, Stem Cells Translational Medicine. So let's welcome Dr. Dopner. I like your shoes.
So, thank you very much. I really do appreciate this award, and I'm pretty sure it will affect my future career as well. Well, being on a stem cell meeting might be pretty provoking to have this kind of title, but since I'm not a stem cell researcher in the first place, but a consultant of neurology, I think I can choose this title. I don't want to talk about the paper that has been awarded, but I rather want to talk about the things that we did back in the day and the things that we want to do in the future. And to start off, I first want to introduce or talk a little bit about stroke. As you all know, or hopefully know, the current concept of cerebral ischemia or stroke focuses on 3 columns. That is the admission of the patient towards the stroke unit, the thrombolysis, and the endovascular treatment paradigm. Unfortunately, these therapeutic options are limited due to side effects and a very narrow time window, and that is the reason why people since about the 1990s have been trying to develop new therapeutic strategies. So the basis of these experimental treatment paradigms is the pathophysiology of cerebral ischemia as depicted there on the slide. And indeed, there were quite a few papers, as a matter of fact, 700, 800 papers using experimental stroke models that could show neuroprotective effects with various drugs. Unfortunately, none of these experimental treatment paradigms have been successful in clinical trials. There's a number of reasons. I don't want to go into details. So the focus has now switched towards studying neurogenesis, and the basis of this is the fact that endogenous neurogenesis persists in the adult brain as well. So as you can see here, there is quite a few stem cell niches, and one of them is the so-called subventricular zone in which the neuroprogenitor cells reside. Upon induction of cerebral ischemia, these cells, these NPCs, migrate towards the site of lesion where they proliferate and finally differentiate into neurons. The problem with this approach is only that both survival rates and differentiation rates of these cells are pretty low. So if you want to use this kind of phenomenon neurogenesis, you have to manipulate these cells. How can you do that? Well, you can protect the NPCs by enhancing the resistance within the ischemic milieu, or what you can also do is you can try to stimulate NPCs per se. And that is, for instance, what we did back in the day by injecting a viral vector that contained a microRNA, in this case the microRNA 122, which induces a neuronal phenotype during ontogenesis of the brain. So we injected this viral vector about 3 weeks before stroke in our mice, and indeed on day 28 and 56 we observed a higher neuronal differentiation rate of these newborn cells. Neurogenesis goes hand in hand with angiogenesis, and in these very same animals, we also observed a higher amount of CD31 expressing cells, thus indicating angiogenesis. So this approach was pretty beneficial for the animals. You could also achieve these very same or similar results by interfering with the afferent B3 pathway. These are proteins that are important during the ontogenesis of the brain as well, especially during axonal projections. And knocking out the afferent B3 pathway also resulted in a higher amount of neuronal precursor cells or neuronal cells within the ischemic lesion site. None of these approaches is really useful for clinical trials. So there's another way how to manipulate endogenous neurogenesis, and this is the transplantation of NPCs. So you can obtain these NPCs from the subventricular zone and put them into the culture dish. They will form neurospheres, and you can easily transplant these cells in mammals. The problem with this approach also is that you have low survival rates and very low differentiation rates of these grafted cells. So again, you have to manipulate these cells. This is a work where we enhance the resistance of these grafted cells by in vitro transduction using TET-HSP70, so a chaperone that will increase the resistance of these future grafted cells. And then we transplanted these cells intravenously or intracerebrally. I don't want to go into detail, but what you can see is, for instance, that when you intravenously transplant these cells, the blood-brain barrier integrity is enhanced when you, for instance, transduce these cells, and also the reperfusion injury in these animals is reduced. When you transplant these cells intracerebral, for instance, what you can observe then is that you have a change within the ischemic micromilieu. In this case, you have increased concentrations of growth factors, which also gives you the hint that these transplanted cells are not really integrated within the residing neural network, but they act in a paracrine way. And that is the reason why we basically came to the approach that has been published in Stem Cell Translation and Medicine. So a few years ago, people thought that the trophic factors, which are secreted by the stem cells, are the decisive mediators, but there is recent evidence that it might be vesicular structures which are more important, or that are the decisive mediators of stem cell actions. So these vesicles are called extracellular vesicles. They can be differentiated into various subvesicles, such as microvesicles, exosomes, and so on. They differ in terms of diameter, origin, and also in terms of contents. And they are virtually secreted by any kind of cell. So we wanted to know, are these extracellular vesicles, these EVs, just as powerful as, for instance, mesenchymal stem cells, or maybe they are not stem cells at all, as we have just heard from Dr. Kaplan? Either way, these stem cells act in an indirect way. So if they act in an indirect way, maybe the EVs will do the job as well. And that is what we started in our stroke model of mice. So in this model, we injected mesenchymal stem cells, or EVs, intravenously on day 1, 3, and 5. And we observed these animals for as long as 28 days. And then we scored them on various time points using behavior tests. And what you can see here is that, first of all, the mesenchymal stem cells do their job. Animals that got the MSCs have a better functional score as the saline control. But also the EVs, or the animals that got EVs are also just scoring as well as the MSC groups. So EVs are not inferior as compared to the MSCs, which is just what we were hoping for. The mechanisms that are involved in this process are not neuroprotection. What the EVs most likely do is they modulate post-stroke immune responses. So this is flow cytometry from blood samples on day 6. what you can first observe is that within the stroke mice, you got an immunosuppression as compared to the sham control. And what the EVs do here is they kind of reverse the post-stroke immunosuppression, as you can see here for the natural killer cells, the T lymphocytes, and the B lymphocytes as well. So this is most likely the way they kind of work. Question now is this feasible in men? Well, we haven't applied EVs in stroke patients yet for obvious reasons, but we were the very first worldwide who actually applied EVs in men at all. And this is a patient that was suffering from a hematological disease, and this patient got a bone marrow transplantation, and unfortunately in the process of this transplantation, the patient developed a graft versus host disease, which can be usually treated using glucocorticoids. In this case, the patient didn't respond to the glucocorticoid therapy. So what you can do then is, in experimental approaches, you can inject or transplant MSCs. We decided to inject EVs instead of grafting MSCs. And these EVs were given continuously for over 2 weeks. And what we then observed was that both the rash and the frequency of diarrhea was significantly decreased. The patient died in about 6 months later, but not because of the EVs, but because of pneumonia. I don't want to say that the EVs actually cured the rash and the diarrhea, but at least we could say that they were tolerated in men. So this makes me pretty optimistic that we can use EVs for stroke patients in the future as well. But before we do that, and this is my last slide, there are many questions to be answered. First of all, we don't really know what is the optimal isolation procedure for EVs. There are many procedures that people can use. Many use ultracentrifugation techniques. We have used precipitation, the so-called PEG precipitation for the project I was just briefly describing. The next question is then, how do we actually characterize these EVs? There's not a single marker which is specific for EVs. And then, of course, we have to further analyze these EVs in terms of therapeutic potential in vitro. And then later on again, we have to further understand what is the real mechanism behind the EV beneficial or the EV-induced outcome. Thank you very much for your attention.